Hello and welcome to the video series of Biotechniques MCQ practice session, and this is session number 2. Again, we're going to practice several important questions, look at the exam corner, analyze the trends, etc. So let's begin with the first question. Which of the following method could be used to determine the structural information about theta DNA? It's a hypothetical one, which you have freshly extracted from a new species of fungus. And the options given here are 1. Cryo-electron microscopy, 2. NMR spectroscopy, 3. electron microscopy, 4. X-ray crystallography, 5. CD spectroscopy, right? So a lot of names are given. Question is all of the above, then all except 3. Question C says option number C says all except 1, and D says 2 and 4. So in order to determine the structure of theta DNA, which is a hypothetical DNA, everything can be used except electron microscopy. So apart from 3, so that means option B is the correct one. Apart from electron microscopy, which has a 10 nanometer resolution, apart from that, every technique can be used. So these kind of questions are pretty popular because in IIT JAM they ask similar type of questions. So all the time they ask something about nuclear magnetic resonance, they ask about X-ray crystallography, circular dichroism. So these names are really popular, and this question was to familiarize you with these names. But anyway, let me tell you how you would get more such questions and how you would analyze the trends. In order to do that, there is a defined batch in IIT JAM, in unacademy for IIT JAM, which is known as Achievers Batch 2022. So if you are preparing for IIT JAM 2022, this batch is for you, and there is only one day to go for price hike. So if you want to join right now and beat the price hike. And in order to join, you can use my code AP10. In this difficult time of COVID, you should study safe and study from home. It is cost effective and time saving. So in order to join, download the unacademy app, use my code AP10, and then you can start learning from the app from the topmost educators in India. Okay, beat the price hike and enroll now. Okay, back to our questions. Before that, I wanted to tell you that with my unacademy course, there are a lot of handwritten flashcards which would really help your preparation. Okay, so now we are still in the exam corner, and I wanted to tell you that there are several questions which talks about techniques like NMR spectroscopy, IR spectroscopy, ESI mass spectroscopy, UV spectroscopy. So all these spectroscopic techniques are super important when it comes to IIT JAM. Not only IIT JAM but other examinations. We would look at it in a moment. So this question is fairly simple. You just need to know the technique, the principle, and then you can answer this question. It was asked in IIT JAM. The technique that involves impacting sample with electron is NMR, ESI mass spectroscopy, IR spectroscopy, and UV spectroscopy. So all of these spectroscopy technique actually use light, but ESI mass spectrometry, that use electrospray ionization technique, so which is bombardment of the sample with this particular electron beam. So that is why ESI mass spec is the correct answer in this case. Okay, then they ask a question from microscopy. So this one particular question and followed by similar questions, we are going to practice to understand what kind of question they can possibly ask from microscopy. So they are not going to ask very difficult question from microscopy. Generally questions are two kind. One is application oriented. Second type of questions are principle oriented. So look at this. Here the principle. So here, which one of the following microscope has working principle? So obviously they are talking about working principle. Most similar to way blind person reads, right? Confocal microscope, epifluorescence microscope, atomic force microscope, total internal reflection microscope. So you just need to know the name of these microscopy techniques and where they can possibly use and what is the key principle, and that's all you need to know for IIT JAM. Here the correct answer is atomic force microscope. The atomic force microscope has a cantilever, and this kind of microscopy is used to understand the surface topology of specific biological specimens or even non-biological specimens as well. Okay, moving your focus from IIT JAM to GAT B, let me tell you that JNU CEB also asks similar questions. For example, secondary structure content of protein cannot be determined by CD spectroscopy, NMR spectroscopy, X-ray crystallography, and FRET, right? So you need to understand FRET tells you about molecular proximity, but it cannot tell you about secondary structure of protein. So all of these techniques can tell you about secondary structure of protein, but not FRET, right? So these kind of questions reminds you that okay, these techniques like X-ray crystallography, NMR, CD spectra, all the techniques that are used in structural biology are very important for IIT JAM. If you want to learn about any of these techniques, the link would be given in the description and also in the I button, so you can click on them, start learning on that part. Okay, then there is a little bit advanced question based on the sequencing techniques used these days. They might ask these kind of questions, but yeah. So the question says match the column A with column B, column 1 with column 2. Okay, RNA sequencing, chip sequencing, ATAC sequencing, Sanger sequencing. So here things are pretty similar that you have faced in other questions. For example, you know Sanger sequencing, you heard about chip sequencing or RNA sequencing, but you have no clue about ATAC sequencing. They would always give some kind of one particular thing that you have no idea of, so don't worry about it. So in this question, this particular stuff is given to confuse you, but don't bother about these things. So always in these questions start with a known factor. For example, you know that chip sequencing can tell you about DNA protein interaction, right? Okay, so it can tell you about it is used to assess the genome wide chromatin accessibility, right? So this is one option, and there is another option that identifies genome wide transcription factor binding sites. Exactly, that is done by chip sequencing. Chip sequencing tells us that where at all in the genome this particular protein or transcription factor might bind, right? So all those sequences would be associated when you pull down that particular protein. So if you want to learn more about chip sequencing, go and click on the link in the I button. Okay. So then RNA sequencing can tell you global transcriptomic profile, right? Yep. So it can tell you about global transcriptomic profile. Now Sanger sequencing can be used to sequence a plasmid. That's true. Like whenever we have a plasmid, we want to know that whether our gene of interest is cloned in the plasmid. You want to sequence the plasmid. The method you use is Sanger sequencing. So these are the thing, right? So S equal to 4, Q equal to 1. So you have to find that S equal to 4, Q equal to 1. S equal to 4, Q equal to 1, and your P equal to 3. So this would be your correct answer. Okay, which of the following is not true about high pressure liquid chromatography, or also known as high performance liquid chromatography? This is very important because people generally avoid this particular topic, HPLC, and people don't have a clear idea about the principle of HPLC. But you should not be the one who don't have that idea. In order to know about HPLC, how it works, click on the I button right now. Okay, it requires high pressure for separation of the species or denilates. There is no need to vaporize the sample. It is performed in columns. It has high sensitivity. So we have to identify which is not true, not correct, or incorrect statement. And the incorrect statement, which is our option, is basically there is no need to vaporize the sample. You would get the detailed description in the I button. Okay. Then there is another question. This question says one researcher identified a unique epitope in his protein of interest. He cloned the sequence of the epitope in an expression vector. Using hybridoma technique, he has generated a monoclonal antibody. But when he used it for detecting the protein in an ELISA assay, he found no signal, right? What could be the reason for this observation? So this is kind of like more TIFR style question or more thought-provoking question. Try to understand. The options are the antibody has very low half-life and it's getting degraded rapidly. Option B says ELISA machine might not be working. Okay, the epitope in India, this is one of the key options, but anyway, the epitope was buried deep in the core of the protein, and as a result the antibody cannot detect it. The antibody was polyclonal instead of being monoclonal. So if you understand that you have taken one particular small epitope, right? So you've taken an epitope, not an entire protein. So you have cloned this epitope in a vector. You put this epitope inside of a plasmid, right? So this epitope has expressed the protein. So this protein is generated, or protein or peptide fragment, I would say. And you have injected this peptide fragment into a mouse, right, or a rabbit, from where you are going to collect the spleen, B cells from the spleen and make the hybridoma. So this particular epitope, but you don't know when you're cloning that epitope that what is the 3D localization of this epitope. So in a particular protein, the epitope might be buried inside the core, right? So then what happens is, if you try to detect that same antigen using ELISA, your antibody might not be able to reach to the core and detect that epitope, and that is why you are not getting any signal. One way of doing it is using some pH change or some agents which can denature the protein structure mildly such that the antibody can bind and give you some response. So this is known as antigen retrieval. So you learned about the concept in this particular question. Okay, last question of the day. This question says match the column 1 with column 2. These kind of questions are very important for IIT JAM and very frequent. Okay, so these are some microscopy techniques, and this question is aimed in a way that it would teach you about the principle. So after solving this particular question, you would get to know what are the principle of these techniques, these microscopy techniques, right? So obviously, let's start. Confocal microscopy, spinning disk confocal microscopy, phase contrast microscopy, atomic force microscopy. And in the column, it is given that monitoring dynamics of fast events such as vesicle transport by kinesin. Option 2 says protein localization inside the cell can be studied using this technique. Then works similar to how blind man reads. It converts phase shift in light passing through a transparent specimen into the brightness changes in the image. So we know confocal microscopy, which works like a pinhole camera, right? So if you don't know what is confocal microscopy, you can click on the I button to understand more about it. Okay, and so this can be used to check protein localization. You can always check my unacademy's Biotechniques course where I have explained microscopy in great details, so you can always refer to that. You can use my code EPI10 to get into that plus courses. Okay, spinning disk microscopy, confocal microscopy is one variant of confocal microscopy which is specially used for high dynamic stuff, like things which are happening very fast in a time. For example, vesicle transport inside a cell or movement, molecular movement, etc. So it has a very high frame rate because it uses multiple pinhole. So for that reason, you are going to use spinning disk microscopy. Phase contrast microscopy. Okay, so here the name tells it all. Phase shift give rise to the contrast, right? So exactly. So the light is passing through the sample, and while passing through the sample, there would be a phase shift. Due to that phase shift, there would be a change in the contrast, and that is why it is known as phase contrast microscopy. And lastly, we have already talked about atomic force microscopy that was asked in some previous year questions, right? So now you know what are the most important thing that you need to learn while you are preparing this particular part, right? These questions answers are really important because it would help you to prepare and help you to understand, okay, I have to spend this much time on this particular concept, how deep I need to go for this concept or not. So in IIT JAM, the overall thumb rule is be superficial but learn a lot. I hope this video was helpful. If you like this video, give it a quick thumbs up. Don't forget to like, share and subscribe. And do let me know in the comment how you like my videos, and click on the bell icon for notifications. Thank you.